amino acids. Again, as I said to begin with, the content that you need to learn across the exam boards does vary for biochem. So please do double check that you're happy with what's expected of you. Read through your specifications and make sure you're confident with what is expected from you. If at any point you're not confident, I would highly recommend going and sitting down with your teacher or a tutor or somebody and going through the specification, because they can help you to understand it and really get your head around it. Hopefully what we've done today has helped, but especially to begin with in the exam technique session. But if you're not feeling confident, or even as you're going through your revision and you're kind of ticking off things on your specification, you come across something you think, I've got no idea what they're asking there, go and talk to your teacher, because I'm sure they'd be happy to help you get your head around it. So with amino acids, with biochem, like I said, there is a variety. So we'll do our best to cover stuff that's across the board, but please do make sure that you're comfortable with what's expected of you. So amino acids are a class of molecules with similar structures. They've all got an amine group, a carboxylic acid group, and then something, an R group. The R group might just be a hydrogen, but it's the R group that determines the chemical properties of the amino acid. So the amine group and the carboxylic acid group will obviously react, or can obviously react, but they are present on every amino acid. So the differences between the amino acids come from what's on the R group. You don't have to know any amino acid off by heart, but I would get comfortable with them in case they come up in an exam. But you shouldn't have to recall them from memory, you should just need to use them. There are 20 different amino acids, which is made using our DNA. And then there are hundreds more found in nature, some of which we do need to consume to make sure that we have got them in our bodies. But they're fascinating. They're lovely. If you are interested, they're lovely to kind of find out more information about. Right. So let's just double check we're happy. The amine group on an amino acid is this bit. That is my amine group. The carboxylic acid group is this. And the ones, I will say, the amino acids that we look at in A-level chemistry, I think you do it slightly differently in A-level biology. If you're a biologist, you do look at extra bits, but we look at what's called an alpha amino acid, which the alpha bit comes from the fact that I think it's from the carboxylic acid. The amine is one carbon away from the carboxylic acid. So I think you can also have, for example, beta amino acids where there's 2 carbons between the carboxylic acid and the amine. So this is in A-level chemistry, just look at alpha amino acids. And then obviously there is that R group as well, and that's what determines the chemical properties. Like I say, it is worth, if you haven't already, it's worth you just having a look at the different amino acids and having a think about what could be interesting or useful about them. So some of them have other amine groups. An amine group can act as a base, has got a lone pair that can interact with things, that can be protonated. Some of them have got sulfurs, which can behave similar to oxygen because they're in the same group. What else? Some of them have got long carbon chains, so would make the species more hydrophobic because long carbon chains are not polar or non-polar, so aren't going to interact with water. So the longer the carbon chain, the more hydrophobic that bit is, etc. Some of them have got alcohols, groups, all this sort of thing. So it's worth having a look and thinking about what kind of questions they could ask you, because that's getting you in the mindset of exams and all that sort of thing. And then you hopefully won't feel too taken aback if you get presented with one in an exam. You might not, but just in case. Okay, so amino acids. Depending on the pH, an amino acid can exist as a positive ion, a negative ion, or what's called a Zwitter ion. Now, this is where there's a little bit of discrepancy. So OCR, I don't think you cover acids in pH at all. Apparently an AQA doesn't need to use what we're going to term as the isoelectric point in a minute. So how does this work? Let's have a look. if you have got a low pH, what color should I do then? Seconds. So low pH. What is low pH? Low pH is acidic conditions. So if there's extra acid around, this is acidic conditions. Low pH. There are going to be lots of H pluses. If there are lots of H pluses floating around, as in you've got a high concentration of H plus, then Le Chatelier's principle says we're going to try and remove those H pluses. We remember acids are in equilibrium with the things that can be protonated. So if we've got a high H plus concentration, anything that can be protonated will be protonated. What can be protonated on an amino acid? The carboxylic acid can be protonated, so it will be protonated, but so can the amine. That means my amino acid is going to have an overall positive charge because everything that can be protonated will be protonated. So it is positive, so plus if, that's me being lazy, positive ion. What about if we've got a high pH? High pH equals very alkaline. Now, this is the reverse. In alkaline conditions, we've got lots of OH minus ions floating around. They will be mopping up any H pluses they can. So again, the equilibrium is going to be lying towards there being very few H pluses in solution. So anything that can try to address the balance and donate H pluses to the solution will, which is why we end up with a carboxylate ion. Anything that can be deprotonated will be deprotonated, so nothing will be protonated in a very high pH. And then if you don't need to be able to explain this theory, but just so that you know it does come from somewhere, the point at which, or the pH at which, an amine will be protonated and a carboxylic acid will be deprotonated is not going to be identical. So there will be a range or a pH, there will be pHs where your amine will be protonated and your carboxylic acid will be deprotonated. And that, if we have a protonated amine and a deprotonated carboxylic acid, we have got a species with two ions, i.e. a Zwitter ion. Zwei is two in German, so I assume that's where it comes from. Zwitter ion, two ions within the same molecule, which is super cool because we rarely have a species which has got two ions in the same molecule. The point at which you have a species with two ions is called the isoelectric point. So that's where this term isoelectric point comes in. Isoelectric point. This is when we have no net charge, we have created a Zwitter ion. No net charge. There are two charges on the molecule, but overall there's no net charge because one cancels the other. Sorry, I should be more specific. The isoelectric point is the pH at which we have no net charge. Let's make that clearer. pH at which, let's not say we, there is no net charge, the pH at which we create a Zwitter ion. So if you're AQA, you don't need to talk about isoelectric point. I think you just need to look at low pH and high pH and what that does to amino acids. Okay, I think that was everything that I wanted to say there. Hopefully that's all making sense. Yes, that was what I was going to say. In an exam, unfortunately, this is the kind of thing which is a little bit scary for students because you do so little on it in your learning and then you're expected to answer a question on it. When you're doing an exam, just look for, if it's a high pH, i.e. an alkaline situation, they're going to have mopped up all the spare H pluses. So anything that, there aren't going to be any H pluses floating around. So anything that can be deprotonated will be deprotonated. So look on the amino acid, whether there is anything that can be deprotonated, such as another carboxylic acid group, or an amine that will keep or stay deprotonated, etc. Similarly, but oppositely, if you've been told about a low pH or an acidic conditions, look for anything that can be protonated. Are there any other amine groups within the molecule that could be protonated? Making sure that all your acid groups are protonated, so aren't ions, etc. Okay, I think that's all I wanted to say. Let's keep going. Excuse me. So, although all amino acids have different chemical properties because of that R group, they can all bond to form the same structure, polypeptides, also known as polyamides. So we've put all there, but it's AKA really. They are also known as polyamides. The way I remember the difference, I think if you're in OCR, you call them all polypeptides. That's what I've got in my notes here. But the way that I've always just noticed the difference is if you're talking about it in a chemical sense, so you're just interested in the reaction, for example, then we call them polyamides because it's an amide bond. If the context is in a biological context, then we call them a polypeptide. That's the difference that I've noticed anyway. So biological context, we'll call them a polypeptide. The chemical context, we call them a polyamide. But just to quell any doubts, they are the same thing. An amide and a peptide bond is the same thing. They are identical. There's no difference. It's just the context. So what is a polypeptide or a polyamide? It is a long chain of amino acids which are joined together by amide/peptide, because they're the same thing, bonds. What are the bonds? The amide bond is the one that we have highlighted in red here. So this is the amide bond, or if it's in a biological setting, it is called a peptide bond. They are formed in a very similar way to the way that we form, let me rephrase that. I consider them to be very similar to the way we form esters. We get the attack of the lone pair of the nitrogen. Oh, let's do that a bit better. Let's do it that way. To the delta positive carbon, you get the movement of electrons up and then back down again, which kicks off the OH. That OH minus ion can then go and steal a hydrogen. The mechanism we've got here is AQA only. So AQA students, you need to know the mechanism. Everybody else, I think it's important to understand what's happening, but you don't need to know the mechanism. You can then see, hopefully, that you get the water molecule lost is as a condensation reaction. Condensation reaction. And the keen-eyed on you will notice that, again, similar to when we talked about the ester bond, I haven't put in all the steps for this mechanism. You would need to show the OH minus ions removing a H plus from the now positively charged nitrogen to form the water molecule that would be formed in the end. Actually, if we've got time, maybe I'll just show that now. I think we have got time. Let's do it. Okay, so what would this look like? This is going to get nicely complicated. What I might do is just show the main bit of the molecule. The bit that we're interested in. So we've got I have not got space for this. Let's do it up here. So we've got our carbon, which has got no, let's do it from there. Sorry, I'm changing my mind all over the place. That nitrogen is attached there. The nitrogen has got 2 hydrogens attached to it. And that has got the rest of that molecule attached to it. I'm going to show what I mean. Nope, I'm using that. So that is all there on that bond, or the rest of that molecule is there, and the rest of that molecule is there. That's what that is at the end. And that's what that is at the end. But I'm not going to bother showing all of it. Otherwise I don't have space actually, to be fair. So then I've got a positively charged nitrogen. How do I know I've got a positively charged nitrogen? Because I have lost an OH minus. Actually, let's stick it up there to make it easier. I've lost an OH minus. That OH minus ion with the positively charged nitrogen means that whole structure is neutral. I started with 2 neutral structures. So when I'm doing my mechanism, I should end up with a neutral structure. Otherwise, I've forgotten to add in a charge, or I've randomly lost some electrons I shouldn't have lost. I can then show, I think I'm going to do these in red actually to make it a little bit clearer. Doop, doop, doop. I can then show the attack of the OH minus ion onto one of those hydrogens. The electrons that belonged to that hydrogen nitrogen bond can return to the nitrogen to resolve that positive charge that was on the nitrogen. Whoops, note pen. And then we can end up, or we do end up with our amine. So that's kind of resolving that middle intermediate stage there. Okay. Was there anything else I wanted to say? Don't think so. No, I don't think so. I think you can also, actually, I will say, I think you can also show the creation of a molecule which has got 2 ions in it. I don't know, I'm definitely not going to have space to do that here, so I'm not going to try. Where you don't get the removal of the OH minus ion immediately, but you get the oxygen holds on to that negative charge. You lose the H plus, or sorry, yeah, you lose a H plus from the amine. The OH minus gains that H plus and then leaves as water as the oxygen kicks that negative charge back down. You can also draw the mechanism out like that. If it's easier for you to remember, that's absolutely fine. It depends whether you've got an acidic solution or an alkaline solution. So, yay, multiple things to remember. Again, though, please do double check your specifications because it may be given to you in only one of the alkaline or acidic conditions. And I haven't double checked that, so I will encourage you to do so.